Hello learners. In our next previous video we talked about asexual reproduction and vegetative reproduction found in Oedogonium. Today we are going to study about sexual reproduction found in Oedogonium, in which we covered the topics related with distribution of sex organs, antheridium, oogonium, fertilization, zygote and germination of zygote. So let's begin. In Oedogonium, sexual reproduction of advanced oogamous type, it is more common in the forms growing in stagnant water than those in flowing streams. It takes place with the help of male and female gametes. Male sex organ is antheridium and female sex organ is oogonium. Antheridium produces anthrozoids and oogonium produces egg. Male and female gametes differ morphologically as well as physiologically. Factors such as alkaline nature of water and deficiency of nitrogenous substances in medium promote sexual reproduction. In some species, it can also be induced by placing mature filaments in an atmosphere saturated with carbon dioxide. Now we are going to talk about distribution of sex organs. On the basis of distribution, Oedogonium are divided into two groups. First one is macrandrous species. Second one is non-androus species. So in macrandrous species, antheridia are borne on filament of normal size. Oedogonium nodulosum and Oedogonium fragile, where antheridia and oogonia are found on same filament, are called as macrandrous monoecious. While other species like Oedogonium crassum and Oedogonium aquaticum, antheridia and oogonia both are borne on different filaments, so they are known as macrandrous dioecious. Filaments bearing antheridia and oogonia are morphologically similar but differ in their physiology. Now second one is non-androus species, in which filaments bearing antheridia and oogonia show morphological distinction, and male filaments that are much smaller than female filaments are called as dwarf male or non-andrium. These species may be monoecious or dioecious. Now next point is antheridium. So monandrous species from terminal or intercalary antheridia by division of antheridial mother cell. Any cell of filament with apical cap can function as antheridial mother cell. This cell divides transversely into an upper smaller antheridial cell and a lower much larger sister cell, which undergoes several transverse division to form a row of 2 to 40 flat uninucleate antheridia. Nucleus of each antheridium divides mitotically into two nuclei. Each gets surrounded by some cytoplasm and metamorphoses into an antherozoid. Each antheridium produces two antherozoids, which is a unicellular, uninucleate and multiflagellate structure. It is similar to zoospore for its smaller size and fewer flagella. So cell wise they are unicellular, means single cell structure and having single nucleus, but many flagella. It is yellowish in color due to its reduced plastids, and their antherozoids are also liberated by transverse splitting of wall of antheridium like zoospores. Antherozoids are also enclosed in a hyaline vesicle at the time of liberation. But in non-androus species, antheridia develop in specialized small 2 to 4 celled filament called as dwarf male, which then develop from androspores formed in androsporangia. While in some non-androus species, androsporangia and oogonia develop on same filament, called as gynandrosporous, and in which both are born on different filaments are called as Oedogandrosporous. Means if oogonia and androsporangia both are developed in same filament, then we are called them gynandrosporous, and if they are born on different filaments, we are going to say them Oedogandrosporous. Now, nucleus of androsporangium does not divide, and protoplast metamorphoses into a single uninucleate multiflagellate androspore. When androspore liberated from androsporangia, it is also enclosed in a thin vesicle. This vesicle gets dissolved and androspores swim freely, sometimes which is attracted chemotactically towards the oogonial mother cell and gets attached to it or any other adjacent cell by its anterior end. Then it germinates to form a dwarf male which has a basal holdfast and one or more antheridia. Nucleus of each antheridium divides to form two anthrozoids, which are similar in structure to those produced by Macrandrus species. Now we are talking about Oedogonium. So development of Oedogonium is similar in Macrandrus and non-androus species. Vegetative cell with apical cap may function as oogonial mother cell, divides transversely into upper oogonium and lower sufflatory or supporting cell. These supporting cell may again functions like oogonial mother cell and form mother oogonium. This supporting cell again function like oogonial mother cell and forms another oogonium and supporting cell. In such cases, two or more oogonia are found in a row. Initially, oogonium is slightly wider than vegetative cells, but at maturity it becomes globous and its protoplast forms a single uninucleate non-motile egg. Young egg is green in color due to presence of chloroplast. At maturity, small pore or transverse slit develops on oogonial wall. Opposite to this slit, hyaline area is present in egg, known as receptive spot. Egg nucleus moves closer to the receptive spot before fertilization. In some species, before fertilization, gelatinous material extruded through the pore to facilitate the entry of the anthozoid into oogonium. So, in our next video, we will learn about fertilization, zygote, and germination of zygote and their life cycle. Till then, happy learning.